Good morning students. In this video we are going to discuss about nucleus and microbodies from the chapter Cell, the Unit of Life. Now let's enter into today's topic in detail. Nucleus as a cell organelle was first described by Robert Brown as early as in the year of 1831. Later to him, the material of the nucleus stained by the basic dyes was given by the name as chromatin, discovered by Fleming. So these two great scientists contributed to the discovery of nucleus and chromatin material. Understood? Now, first I would like to tell you one important point here. The interface of nucleus. When the cell is in interface, it won't divide furtherly into two dotted cells. The interface is also called as rest phase or resting phase. So when the rest phase nucleus is highly extended and elaborate, nucleoprotein fibers are called chromatin, and nuclear matrix and one or more spherical bodies are called nucleoli, singular nucleolus. Understand? You can see on the diagram structure of nucleolus which is present on the screen. So electron microscopy has revealed that the nuclear envelope, as I told you, electron microscope usage have started in the decade of 1950s. So in this decade only, detailed explanation of plasma membrane, ribosomes, nucleus, etc. everything came from light because of the invention of electron microscope. So according to electron microscopic structure, the nucleus have covered with envelope called as nuclear envelope. So nuclear envelope has two membranes, outer nuclear membrane and inner nuclear membrane, which run parallel to each other with evenly spaced. So the space which is present between outer nuclear membrane and inner nuclear membrane is called as perinuclear space. So the space is about 10 to 50 nanometers. Understand? So this is very important. Please make a note. Next, this perinuclear space forms a barrier between the materials present inside the nucleus and out of the cytoplasm. So the outer membrane usually remains continuous with the endoplasmic reticulum. So the outer membrane which you are seeing on the diagram, the outer membrane is continuous with the endoplasmic reticulum, and since the endoplasmic reticulum possesses ribosomes on it, you can see ribosomes which are red color in this diagram. So you can also see outer nuclear membrane and inner nuclear membrane, both membranes together called as nuclear envelope, and this outer nuclear membrane exclusively connected with the endoplasmic reticulum, especially rough endoplasmic reticulum. Rough endoplasmic reticulum possesses ribosomes. These ribosomes are meant for synthesis of proteins. Hence, these ribosomes are also called as protein factories. At a number of places, the nuclear envelope is interrupted by minute pores. You can see at different places the nuclear envelope got disturbed and formed small pores. Those pores are called as nuclear pores. You can see nuclear pores on the diagram right now. Next, what is the role of these nuclear pores then, which are formed by the fusion of its two membranes? So nuclear forms are forming by the fusion of its two membranes. So both inner and outer nuclear membranes are fusing together and interacting at some intervals of the nuclear envelope and forms a small pores. Those pores are called as nuclear pores. So the nuclear pores are the passages here, helps as a barrier. So the nuclear pores are the passages through which movement of RNA and protein molecules takes place in both the directions, between the nucleus and the cytoplasm, from the cytoplasm to nucleus, vice versa. So all the materials or molecules move through these nuclear ports only, from nucleus to cytoplasm, cytoplasm to nucleus. So it acts as a barrier. Next, one more point I would like to tell you here. Normally there is a one only nucleus is present in a cell. So mononucleus condition, it is called as mononucleus condition. You can see mononucleus condition in the cells generally. So here one more point here, when we can also see variations in the number of nuclei in different cells, frequently observed in many cells. Understand, I would like to tell you one important neat point here. uh, Number of nuclei are vary from one cell to another cell. It depend upon type of the cell. Understand. Next, some mature cells even lack of nucleus. It is one amazing point and interesting point. Okay, let me say examples. Erythrocytes are also called as commonly red blood cells. So the color causing pigment is hemoglobin, so they are red in color. So these erythrocytes of the many mammals like human beings and sieve tube cells of vascular plants. So especially matured cells, not young cells. Matured cells of matured RBC cells of many of the mammals and sieve tube cells of vascular plants are not possessing nucleus. They are called as enucleate cells. They are called as enucleate cells. So cells which possesses nucleus, they are called as nucleate cells. Cells which don't possesses nucleus, they are called as enucleate cells. So example for enucleate cells: matured erythrocytes of many mammals and sieve tube cells of many vascular plants. Vascular plants means plants which possesses vascular tissues like xylem and phloem. Those are called as vascular plants, like mango, neem, tamarind, eucalyptus, etc. All these comes under vascular plants. So in these vascular plants, sieve tube cells which comes under phloem tissue do not possesses nucleus in them, hence it is called as enucleate cell. Understand? Hope you understood this explanation. Now I am going to explain the continuation concept. Please watch the diagram. So the nuclear matrix, it is also called as nucleoplasm. The nuclear matrix, it is also called as nucleoplasm. The light pinkish color structure which you are seeing on the screen right now, it is nucleoplasm, contains nucleolus. So inside nucleolus, spherical nucleolus structure is there. You can see nucleolus and chromatin. So which are chromatin here? The fiber like structures you can see here in this diagram. So the reddish fibers or network or filamentous structures are called as chromatin. So generally nucleus consisting spherical nucleolus, plural nucleoli, and the fibrous or filamentous or thread-like structures called chromatin. And one more point here regarding this one: the nucleoli are spherical structures present in the nucleoplasm. The content of the nucleus is continuous with the rest of the nucleoplasm. Understand, as it is not a membrane bound structure. So the nucleolus, which is blue in color here, it has some content. The content is joining or mixing with the nucleoplasm. Understand, since the nucleoli is not covered with any kind of membrane, the content which is present in the nucleoli is freely mixing or moving with the nucleoplasm which is present in the nucleus. Understand? Then this nucleolus is playing one important role. What is that role here? The nucleoplasm is not surrounded by any kind of membrane on the surface, and it is the site for active ribosomal RNA synthesis. This is most important for NEET examination. So the nucleoli is the site for active ribosomal RNA synthesis, shortly called as R, small r, capital RNA. It stands for ribosomal RNA. Understand? So which is the site for synthesizing active ribosomal RNA in a cell, it is nothing but nucleoli. Then larger or more numerous nucleoli are present in the cells actively carrying out protein synthesis. So larger or more numerous, it means many in number, nucleoli are present in the number. Understand? So they are playing a great role in here synthesizing of proteins, carrying out synthesizing the proteins. Understand? So I wanted to tell you one more important point here. You may recall that the interface, what is meant by interface? Just I said at the time of introduction, interphase is a phase where the cells are not dividing furtherly or any kind of changes are not occurring when the cells are in interphase. So it is said to be as rest phase or resting phase. Understand? When the cell is in rest phase, if you see the nucleus has a loose and indistinct network of nucleoprotein fibers. I repeat once again, what kind of structures those are? Loose and indistinct. It means not clearly visible. Distinct opposite indistinct. Network of nucleoprotein fibers are called as chromatin. So chromatin structures, just now I showed you here in this diagram. Once again you see it, they are in red color. Understand? But during different stages of cell division, cells show structured chromosomes. So you can see one structured chromosome here. So during different stages, when the cell is undergoing different stages, so the chromatin material changes into structures like chromosomes in place of the nucleus. In place of the nucleus. Understood? So chromatin contains DNA and some basic proteins called histones. So the chromatin consisting, what are those? DNA, deoxyribonucleic acid, and some basic proteins, they are called as histones. They're called as histones. This is very important for NEET. Please remember, make a note it. Then apart from DNA and proteins histones, some non-histone proteins also can be seen, and also RNA molecules also can be seen. So this is most important for a NEET exam. So what kinds of materials can be seen here? The chromosomes which are present in the nucleus. So the chromatin consisting what are those? DNA, deoxyribonucleic acid, and some basic proteins called histones, and some non-histone proteins, and also RNA molecules. First of all, I wanted to tell you how a chromosome is forming. Here you can see on the diagram, two chromatids are present side by side, light together and formed as a single chromosome. Understand? So one chromatid thread which is a proteinous filament or nucleoprotein fiber, it is joining with another nucleoprotein fiber and forms as a single chromosome. So here the chromatin consisting DNA and proteins called histones and non-histone proteins and few RNA molecules. Understand? A single human cell has approximately 2 meters length of DNA. What this is also important, how many meters of length of DNA can be seen? A single human cell has approximately 2 meters long thread of DNA distributed along its 46 chromosomes. 46 chromosomes means 23 pairs of chromosomes. In those 22 pairs are autosomes, one pair is allosomes. Autosomes means non-sex chromosomes, allosomes means sex chromosomes. So here you will study in detail regarding DNA package and the function of chromosomes and the formation of chromosomes, all these things in the another further chapters. Understand? So here I wanted to tell you one important point here. On the screen you are seeing one chromosome. Understand? Let us discuss about this one. Every chromosome essentially has a primary construction. So the primary construction is also called a centromere. So you can see centromere here, which is present in the center. So every chromosome has a primary construction. whatever the chromosomes are present, every chromosome is having primary construction or centromere on the either sides of which disc-shaped structures called kinetochores. Either sides, you can see both the sides, the yellowish structure which is disc-shaped called as kinetochores. So how many kinetochores can be present in one chromosome? Two kinetochores. So each chromatid has how many kinetochores? One kinetochore, right? So since one chromosome has two kinetochores, then where the location of centromeres, where the disc shaped structures kinetochores are present, at that the primary construction centromere is present. So you can see here centromere region of the chromosome, either sides yellowish kinetochore structures which are disc shaped can be also seen on the diagram. Next, based on the position of the centromere, the chromosomes are generally and broadly divided into different categories. There are four in number. How many types? Four types in number. So there are four types of chromosomes you can see on the screen. First one metacentric, second one submetacentric, third one acrocentric, and last one coelocentric. Now let us discuss about each one. First, metacentric chromosome. So make sure one point must be clear. All these four types of chromosomes are divided based on the centromere position. You can see centromere position red in color in these chromosomes. Now watch the centromere structure and position very clearly where it is present. So first I would like to explain about metacentric chromosomes. The metacentric chromosomes has middle centromere forming two equal arms of the chromosome, upper arms and lower arms. So exactly to the center of the chromosome centromere is present. So due to presence in the center of the chromosome, the centromere is dividing the chromosome into two equal halves. So upper arms and lower arms are equal in size in this metacentric chromosome. Next, second one, submetacentric. In the submetacentric chromosomes, the chromosome has centromere slightly away from the middle of the chromosome. So you can see in this diagram, second diagram, submetacentric chromosome. So the centromere is away slightly from the middle of the chromosome, resulting into one shorter arm and one longer arm. So you can see towards upside shorter arms, towards downside longer arms. So how this difference is forming? Because slightly the centromere is away from the middle of the chromosome. Understand? These type of chromosomes are called as submetacentric chromosomes. Next, third one, acrocentric chromosomes. So in acrocentric chromosomes, what kind of nature can be seen? So in chromosome which is having centromere exactly very close to the end of the chromosome, then the chromosome's arms are upper arms are extremely shorter, lower arms are extremely longer. Understand? Acrocentric chromosome, the centromere, in the acrocentric chromosome, the centromere is situated close to its end, forming one extremely short end and one extremely long end or arm. Understand? You can see on the diagram, one end is extremely shorter, one end is extremely longer. So upper arms are extremely shorter, lower arms are extremely longer. Understand? Last one, telocentric. Whereas the telocentric chromosomes has terminal centromere. You can see centromere exactly at the end or at the top or the tip or terminal position of the chromosome. Understand? So we can't identify upper arms, we can identify lower arms very clearly, which are large in size. Understand? These are the four different types of chromosomes. Once again I repeat, metacentric, submetacentric, acrocentric and telocentric. So this is very important question for PU board theory examinations. Explain different types of chromosomes based on the position of centromere. In such a case, when they ask this type of question or this question, you need to explain in detail with 200 to 250 words and with detailed label diagram, then only you can get 5 out of 5 marks in PU board theory examinations. Understood? Now students, the same types of chromosomes you can see on the screen very clearly. The first one is metacentric, second one is submetacentric, and third one is acrocentric, and last one is telocentric. Understood? So these are NCERT textbooks. Please draw it. Next, students, I wanted to tell you the last cell organelle in this chapter. Sometimes you know what happened, few chromosomes have non-staining secondary constructions at the constant location, and this gives the appearance of the smaller fragment called as satellite. So satellite structure you can see in this diagram, first diagram in the metacentric chromosome. So how satellite is forming? Sometimes a few chromosomes have non-staining secondary constructions at a constant location. At a constant location means always towards up, at the end of the upper arms only. So they are non-staining ones, so can't be visible very clearly, can't notice very clearly. The two not in all chromosomes, in few chromosomes only. Understand? So sometimes a few chromosomes have non-staining secondary constructions at the constant location in the bracket, or at the tip side, at the tip location. Those are gives appearance as a small fragment called as satellite. So what is meant by satellite? Satellite is nothing but a small fragment which is present at the tip or terminal position of the chromosomes, which is non-staining the secondary constructions. Understood? So microbodies, coming to microbodies, students, microbody structure you can see on the screen. Microbodies are having different organelles in them, like glycosomes, glycomeres, and peroxisomes. All these together called as microbodies. Since they are micro in size, they are called as microbodies together. So microbodies includes peroxisomes, glycosomes, these three. Please make note, this point is out of the NCERT textbook, but very important for knowledge to know. Understand? So what are those? I wanted to repeat: peroxisomes, glycosomes, and glycomeres. All these three together collectively called as microbodies. So many membrane bound minute vesicles. What are those? Minute vesicles presence in the cytoplasm of the cell, they are called as microbodies, and they contain various enzymes. Understand? So they are present both in plant cells and as well as animal cells. So they are enucleate one, nucleus is present, and mitochondria is also present. They are present both in plant cells and animal cells. So microbody structure you can see on the screen. They are present on the body in different plant cells and animal cells. They are small microbodies and vesicular structures, number of minute structures present in the cytoplasm, and their role is producing various kinds of enzymes. Understand? Students, hope you understood this video. I conclude this chapter with these microbodies. Thank you one and all.